Dear students, I am Sher Lakho, lecturer Government Degree College Kandiaro. My topic is nerve impulse. I am lecturer of Zoology. My subject is Zoology. I have selected my topic from the chapter number 3, class enter. Topic is nerve impulse. Now, first of all, what is nerve impulse? Definition of nerve impulse. Now, nerve impulse is electrochemical message. I am writing here. Nerve impulse is electrochemical message. Electrochemical message from one neuron to another neuron or wave of electrochemical message which passes from one neuron to another neuron. Suppose I have two neurons. Neuron number 1. This is neuron number 1 and this is neuron number 2. This is neuron number 3 and neuron number 2, neuron number 3. How the impulse travels from one neuron to another neuron, we have to discuss it. This is called nerve impulse. Nerve impulse simply, it is impulse of electrochemicals. In this, electrons as well as chemicals, they are involved. There is wave of electrochemical message travels into the nervous system. How nerve impulse is developed? It developed by 4 ways. First of all, we have to study resting membrane potential, then we have action potential, then we have propagation of impulse. Number 4th is synapses. Number 4th is synapses. Now, number 1, resting membrane potential. Here the word indicates membrane means membrane of the neurons. The membrane of neuron is called neurolemma. The membrane of neuron is called neurolemma. If we see the neurolemma, the charges are the potential difference of the neurolemma at the resting condition. It means there is no stimulation or there is no external change takes place in the neurolemma or in the neurons, then this type of condition is found. This is outer region of neuron and this is inner region of the neuron. The outer region having a positive charges and inner region having a negative charges. The outer region, it also contains sodium and inner regions contain many different organic chemicals. That's why there is potential difference, and in this condition the neurolemma is called polarized. Now this neurolemma is polarized neurolemma because positive and negative charges are present here. Scientists have measured the potential difference of human neurolemma is about -65 millivolt. It means during resting condition or during unstimulation or during no flow of message or during no flow of excitement or during no flow of waves in the neurons, then the potential difference is minus 65 to minus 70 millivolt. So then we have action potential. Now action potential during stimulation, what happens? The sodium here, sodium is greater than greater in outside, and potassium is greater in inner side. What happens during stimulation? During action potential, sodium enters into the neurolemma, and potassium goes out of the neurolemma. It means depolarization and repolarization takes place. How repolarization and depolarization takes place? This positive charge enters into inner side of the neurolemma, and negative charge comes out, and the negative charge is developed in the neurolemma. In this way, the message or impulse is developed, and I have represent here depolarization and repolarization. Depolarization means when sodium enters, and when sodium enters, then there is the polarity of the neurolemma changes. When the polarity of the neurolemma changes, then the impulse flows from one neuron to another neuron. This flow of the charges, this flow of the electrochemical message is called nerve impulse. Now this membrane or neurolemma is in action potential, so the potential difference changes during resting membrane potential or during RMP. It is also called RMP. The potential difference is minus 65. During action potential, during depolarization, the potential difference changes. It changes up to 40 millivolt. So action potential is depends on depolarization and repolarization. Now propagation of impulse. How impulse travels? Impulse travels due to change in the polarity of the neurolemma, and the sodium and potassium pump develops. Sodium enters inner side of the neurolemma, potassium comes out of the neurolemma. In this way, the polarity changes, and there is difference develops in one neuron. Then this neuron travels to another neuron, then this impulse travels from one neuron to another neuron, and this is called propagation of impulse, or it is also called transmission of impulse. This is a one neuron, and this is a second neuron, and this is third neuron. In this way, the impulse from one neuron, it travels to second neuron, from second it travels to third neuron, so on, and this process takes place. Now, how this propagation of impulse occurs? We are going to discuss it. How this occurs? There is a process that is called synapses. Synapses develops. How synapse develops? Now I am writing here synapses. Keep in your mind that the neuron is composed of neuron consists of three regions. Neuron, how many regions? Neuron consists of consists of three regions. One is called dendroid, other is called soma, and third is called axon. Now what happens? This neuron, there is a neuron number 1, this is a neuron number 2, neuron number 1 and neuron number 2. Now how synapse develops? Synapse is this is called this fusion of two neurons is called synapses. When two neurons they fuse, they remain together for what purpose? For the electrochemical message. This is called synapses. Keep in your mind that synapses always takes place between the axon of the one neuron and dendroid of another neuron. Here, this is called axon. This is called axon, and this is called dendroid. This is neuron number 1 and neuron number 1 and neuron number 2. This region is this is called axon, and this is called dendroid. This cleft, this region is called synaptic cleft. This region is called synaptic cleft. How impulse travels from one neuron to another neuron? It travels, suppose, see here, impulse travels from here, from one neuron, then it will travel to another neuron. It means it will travel from neuron number 1 to neuron number 2. How it occurs? When the polarity of the first neuron is changed, then some type of changes also occur. The calcium is released. In the axon of first neuron, there are some vesicles. These vesicles, these are the bags. They have some neurotransmitters. It has some neurotransmitters. When calcium is released from the T channels of the neurolema, the calcium will bind with the calcium ion will bind with the vesicles, and neurotransmitters, they will move to the receptors. These receptors are present and the dendroid of another neuron, and in this way the transmission or propagation of impulse will occur, and this joining of the two neurons is called synapses. Synapses consist of three regions. First region is called presynaptic, and second, this is called small type of cleft is called synaptic cleft, and the dendroid of the other neuron is called postsynaptic membrane. Now there are many neurotransmitters. When these neurotransmitters they travel from the vesicles, how they travel? Due to combination with calcium ions, they will be attached at the receptors and the dendroids of another neuron. Then there will be new impulse, or there will be new electrochemical change. There will be new potential difference develop in this neuron, and in this way nerve impulse is developed.